core to the cloud. Perfect. Yep. We're doing
Give
good.
me 1 second. Somehow I got multiple windows open. All right, sharing. And I'll go back into presentation mode. All right.
Okay, we're good. You're there.
Looks good.
Melody, can you see that Walter Wynn is on Zoom? Let's see.
Let me look. I do
I-
see Walter.
Yeah, there he is. Okay. About ready, Melody?
Yes.
Okay. We'll go ahead and get started. Good morning. Thank you for coming. If you're in the room and you have a cell phone, if you could please silence it, it'll be greatly appreciated. For those on Zoom, you are muted. Please stay muted. You can ask any questions in the chat after the presentation, and I'll be monitoring the chat. Walter Wynn is here, and he's going to introduce our speaker who's on Zoom today.
Good morning, Melody. Melody was born and raised in Hollywood. She received both her bachelor's and master's degree in entomology and nematology from UF. Currently a first-year PhD student in the Vector Entomology Lab at UF IFAS in Fort Lauderdale. She's working on field-capable portable multiplex assay devices, if I got that all right. So Melody, welcome, and you may start at any time you're ready. Thank you.
All right. Good morning, everyone. As you just heard, my name is Melody Bloch. I'm an entomology PhD student at the University of Florida, and I am located at the Fort Lauderdale Research and Education Center. My research focuses on the use of environmental DNA, or eDNA, for the detection of various invasive reptile and fish species. Today, I will be showing you how my lab developed a multiplex digital PCR assay for eDNA monitoring of 4 invasive snake species in Florida. And this is part of my dissertation. So to start off, invasive species are really prevalent here in Florida, and that's due to a number of factors. For one, Florida has a subtropical climate. It's nice and warm here, and that makes it an ideal place not only for the tourists, but also for these invasive cold-blooded animals such as insects, reptiles, and fish. Florida also has a peninsular geography and disturbed habitats. And due to those factors, there is an absence of these natural competitors. So the invasive species are able to exploit these ecological niches, and they have a wide availability of resources for them. We also have a thriving exotic pet trade here. So invasive species can be introduced from that pet trade either due to accidental escapes or intentional releases. And there's also major ports of entry here in Florida, which also increase the probability of these invasive species being introduced. Now, the invasive species that establish here are successful because they have some characteristics that allow them to properly establish here, and those characteristics include high reproductive rates. For example, in the figure to the left, this is a picture of a Burmese python clutch. So the Burmese python is a rather infamous invasive species here in Florida, and this is a picture of its clutch. And these clutches can typically contain around 50 to 60 eggs, but it can range anywhere from 10 to even 80. They also have a really long lifespan. They are capable of dispersing wide distances, and they're also very adaptable. So some of these invasive species are very cold tolerant. For example, there's some invasive species of fish that we have, including the Asian swamp eel, that is able to tolerate colder temperatures. So it might start out here in Florida, South Florida, and spread further distances where it's colder. They also have a very general diet. So these constrictor snakes will they will eat whatever they can physically take down. They're not very picky. So that makes them really adaptable to South Florida. And they have a huge impact here. For one, they interfere with the Everglades restoration efforts. So in the figure to the right, you can see the natural flow of the Kissimmee River, and Lake Okeechobee is shown here. And you can see that natural flow in the middle is what the current flow looks like. And on the right is the plan for the Everglades restoration efforts to make that flow more similar to that natural flow. And these efforts are being made to improve Florida's water quality and also protect buildings and homes from flooding. Now, the invasive species interfere with those efforts. They also threaten and feed on native wildlife. The invasive fish that I talked about earlier can threaten some crayfish that are really vulnerable species. They also introduce parasites to wildlife. So going back to the Burmese python example, these Burmese pythons contain a lung parasite. It's called the snake lungworm, and this is a crustacean parasite that is from the snake's native region. So in Asia, this snake has been exposed to this lung parasite, and it's actually introduced that parasite over here in Florida. Now, because this parasite is from the snake's native homeland, it has had time to evolve with this parasite. It's had this constant exposure to the parasite, so it is pretty tolerant of it. And it also, the Burmese python is also just rather large, so it can tolerate these parasites pretty well. But the problem is, with the Burmese pythons here in Florida, they've actually introduced the parasites to our native snakes. So now the researchers are finding these parasites in the lungs of the native snakes, and these native snakes have not been exposed to this parasite before, so it could be really harmful for them. The native snakes are also much smaller than the Burmese pythons, so it harms them even more so. Invasive species also increase the prevalence of zoonotic viruses. And the way this happens is the constrictor snakes, for one, will feed on large mammals such as deer, decreasing the populations of those large mammals. And so the local mosquito population will feed more often on smaller mammals. And these small mammals actually contain an Everglades virus. So by the constrictor snakes more often feeding on the large mammals, decreasing those populations, the mosquitoes feed more often on the small mammals, and this increases the prevalence of that virus and the spread of that virus. So clearly, there's a variety of problems that come with these invasive species, so we need to be able to do something about them. So in order for us to manage these species, remove them when possible, and eradicate them when possible, we need to first know where they are. We need to find them, we need to monitor them. And there's a couple typical monitoring methods that are used right now, and these include visual surveys and radio telemetry. So the visual surveys are exactly what they sound like. That entails going out into the field, typically on an airboat, and looking for these snakes, oftentimes at night because they are nocturnal, so they are most active at night. And radio telemetry. Now with the radio telemetry, this is a little more labor intensive. So they will actually capture a Burmese python, surgically implement a radio transmitter, release that python with the transmitter into the field, and then they'll actually use a radio to track it down in hopes that that Burmese python will lead the researchers to other Burmese pythons during their mating seasons. Now, these two methods of monitoring have a really low detection probability. There's a study demonstrated that it's only around 5%, and that's due to a number of factors. For one, these constrictor snakes are really cryptic. They can blend in really well, and they can hide really well. And you would think that a Burmese python that can be around 10 to 16 feet in length would be easy to spot, but it's not. And that's due to the snake has this coloration that allows it to blend in with the vegetation and the water really well. And two, they can hide in that vegetation. So on the left is a figure of Burmese pythons. All of these white circles here show where the Burmese python is hiding. And you can see it's really hard to spot them out in the field. And this is in the middle of the day. Imagine looking for these animals at night. So they can hide in the vegetation really well. They can hide in the water really well. They also experience periods of inactivity. So it's hard to find them when they're not moving around much. And they dwell in areas that are hard for people to access. So like I said earlier, the researchers, when they go looking for these animals, they're taking airboats, they're looking for them at night when they're most active. But they often are dwelling in places in the Everglades that are really remote, really hard to access, and in vegetation that is hard for people to get through. So essentially, we need to improve the way that we monitor for these animals, look for these animals. And one possible way to do that is with environmental DNA. So this is DNA that is obtained from substrates. And the way this happens is animals, as they interact with their environment, they're constantly shedding DNA. So these different substrates that you can get environmental DNA from include air particles. If the animal is aquatic or so much as drinks or uses the bathroom in the water, you can sample the water and find their DNA. If they interact with soil at all, we can find it in the soil. If they interact with plants at all, if they step on a plant or rub up against a plant or crawl on a plant, we can actually swab the surface of plants and get eDNA from that as well. You can detect eDNA from fecal matter or even stomach contents as well. And this is essentially a way to monitor these cryptic species without having to physically see them. We compare this eDNA with digital PCR, which is a form of PCR that functionally replicates a reaction over 20,000 times, making it really highly sensitive to these small amounts of DNA. And the digital PCR system that we use allows for multiplexing, so the detection of multiple targets. In our case, we're using it to detect multiple species at a time. And it also allows for absolute quantification of the target section of the DNA. So by pairing the environmental DNA with digital PCR, it allows for rapid and strong detection of these traces of DNA from field samples. So before I get too much into the methods, I first wanted to introduce you to our 4' suspects for this assay. So these are all nocturnal, semi-aquatic constrictor snakes that have been established in South Florida. The first is the boa constrictor, shown here. It is from South America, and it's been established and breeding around the Charles Deering Estate in Miami since probably around the 1970s, although the exact time point is not completely known. And they feed mostly on rodents and opossums. There's the Burmese python, the infamous Burmese python, which is from Southeast Asia. And these have had a widespread, very widespread established population in the Everglades in South Florida since the 1980s. And they have a high impact on small mammal populations. Now, they do eat deer and birds, and very occasionally they will eat alligators, but they are really strongly associated with declines in raccoon, opossum, bobcat, rabbit, and fox populations. There is the northern African rock python, shown here. This one goes by a few different common names. It's also known as the African rock python or the Central African rock python, and that's to distinguish it from the Southern African rock python. Now, this one is from sub-Saharan Africa, and it has a very small established range in Miami that's been known since the 2000s. And they also feed on a wide range of mammals and birds. And finally, there is the rainbow boa. This snake is from Central and South America, and it is the most recently established constrictor snake of these 4. And it's been around here since at least 2024, with a known established population in Homestead. And they feed mainly on rodents, but also other small animals, such as birds, even eggs, lizards, etc. And this is the smallest size constrictor snake of these four. It's only 4 to 6 feet long, whereas the others can get to between 10 and 16 feet long as adults typically. Now, I just wanted to point out that this picture is not Photoshop, it's not AI or anything like that. The rainbow sheen that you're seeing on here is actually natural for this snake because it has this structural coloration on its body, hence the name rainbow boa. I think it's really neat. Now, for the methods of this project, we included multiple forms of PCR. So we're functionally replicating the DNA target to detect these different species of snakes. Now, with conventional PCR or standard PCR, this is essentially a way to assess presence or absence of these species. And we know that we have a positive when you see these white bands on an agarose gel. If there is no band, that means it is negative. With quantitative PCR, this utilizes a fluorescent tag typically. And so as the PCR cycles occur to make these copies of the DNA, more fluorescence is occurring, and the software essentially detects that fluorescence over the PCR cycles over time. So the results of a positive PCR reaction for the quantitative PCR looks like this, this graph with an increasing line here. Now with the digital PCR, this also uses fluorescence for the detection, but instead of just one PCR reaction occurring, it is, like I said before, over 20,000 reactions occurring. So this is essentially just positive versus negative, presence, absence, but replicated over 20,000 times. So for the primer and probe design of this assay, we wanted to make sure that we were able to detect all 4 species of these snakes, but we wanted to make sure we could actually distinguish between them using this assay. And we wanted to minimize any risk of false positives or false negatives. So we designed unique primers and probes for each of the 4 species. And these primers and probes contain regions of the DNA that are unique to each species. They have a number of SNPs or single nucleotide polymorphisms that make them unique to their respective species. So we also included some native species of snake when we designed these primers and probes, just to make sure that they wouldn't get detected and they wouldn't lead to any false positives. So once we received our primers, we then tested them using snake muscle tissue DNA extract for standard PCR. And we assessed a number of different annealing temperatures. So the temperature at which the primers bind to the DNA. We checked a range of these temperatures to see what would work best. And you can see these primers work with our positive controls at a variety of these different temperatures shown here. So at this point, we were confident that our primers work, but we needed to make sure that our probes also work. So then we moved on to quantitative PCR. So at this point, we are testing our primers and probes together for each individual species. And we also tested the assays on the species that do not correspond with them to make sure that there is no cross-amplification, no false positives occurring. So these numbers shown here are the CT or cycle threshold for the quantitative PCR. That is essentially the number of cycles that it took for the fluorescence to surpass a certain threshold. That number will appear when the DNA amplifies. It will not appear if the DNA does not amplify. So you can see based on this table that the boa constrictor assay, the primers and probes, only work for the boa constrictor. Same thing with the rainbow boa, the North African python, and the Burmese python. And here is the graphs of this result. So these are the amplification charts. You can see the increase in fluorescence for the target species, but everything else is this flat line. It did not amplify for all 4 species. So at this point, we were confident that our primers and our probes work together and that they don't cross-amplify the other species that we're trying to target. So next what we did was we took the standard PCR products and we cloned them and obtained plasmids from them. So this was a way for us to isolate just that section of the DNA that we're copying or amplifying. And then we serially diluted those plasmids, starting with 10 to the 7 copies per microliter down to 10 to the 1 copies, or 10 copies per microliter. And we also did this with total DNA samples. So the muscle tissue DNA extract, we diluted it to 25 nanograms per microliter and serially diluted it down 10 to 1 to 0.025 nanograms per microliter. And we did this to test our digital PCR to see at what concentration is ideal for further testing when just that DNA section is isolated or when we're using total DNA. And also, what is our limit of detection with both of these forms of DNA? So these are the results of the digital PCR with just the primers and probe for North African python tested. And the different concentrations are shown here. So you can see at 10 to the 7 copies per microliter down to 10 to the 4 copies per microliter, the digital PCR percent positives are either 100% or very, very close to 100%. So that means that of those 20,400 wells, all of them are testing positive for this snake, for this snake's DNA. Now, we determined that we considered this oversaturated because for the digital PCR to absolutely quantify the number of copies in that sample, it uses a Poisson statistical model. And for that model to work properly, there needs to be at least 1 negative in those 20,000 wells for the math to work properly. So we considered these oversaturated because of that, and we wound up deciding that our ideal concentration is 10 to the 2 copies per microliter, which had around 10% positive wells. But we were still getting noticeable positives, even with just 10 copies per microliter of a sample. And for the total DNA samples, we see something similar happening. When the concentration is 25 nanograms per microliter or 2.5 nanograms per microliter, we're getting oversaturation. So our optimal concentration for further testing was 0.25 nanograms per microliter, which had around 32% positive wells, but we are still getting noticeable positives with the concentration of 0.025 nanograms per microliter of total DNA. And here is a visual representation of these results. So these are the chip images or array images of the digital PCR. So in this square here, all of those teeny little pixels or dots are those 20,400 wells. And anything that is blue is positive. So you can see the the plasmid that had 10 to the 7 copies per microliter, all of these wells are positive. With 10 to the 2 copies per microliter, there's a noticeable amount of positives, but some negatives. And with 10 to the 1 copies per microliter, or just 10 copies per microliter, there's still some positives you can sort of notice, but it is mostly negative. And same thing with the total DNA. So with 25 nanograms per microliter, everything is positive. With 0.25 nanograms per microliter, still getting a noticeable amount of positives, but some negatives. And with 0.025 nanograms per microliter, it is still clearly, we are still clearly getting positives out of this sample, but it is mostly negative. So then we actually moved on to our multiplex assay. So up until this point, we were using the primers and probes for each species separately. We were creating different mixes to just test those primer and probe sets on their own. And so at this point, we obtained our multiplex assay and wanted to test it. Now that we know that the primers and probes work separately, we want to see if the multiplex assay works when the primers and probes are all mixed together in the same liquid. So to do this, the way that it works in the digital PCR is we have to assign different fluorescent tags to each probe. And that way they light up as different colors. So for the boa constrictor probe, it was assigned June, which lights up red. For Burmese python, we assigned it the fluorescent tag Abby, which lights up yellow. For rainbow boa, Vic, which lights up green. And for North African python, blue, which is the name FAM. Now, at this point, we wanted to, again, ensure that there is no cross-amplification occurring. So we tested these with our muscle tissue positive controls at the optimal concentration that we determined earlier, and also our plasmids. And we found that, again, the boa constrictor assay only amplifies boa constrictor. You can see these are the chip images of the digital PCR, and only the boa constrictor was positive in this sample. Same thing with Burmese python, working with the Burmese python assay, rainbow boa, and North African python again. Anything else was negative, which is what we want. And here are some graphs of, again, the same digital PCR data with the total DNA samples. So when looking at these graphs, any of these gray dots down here are considered negative because they're below the threshold, the fluorescent threshold. Anything above that threshold is a colorful dot, and that represents the positives. So with these graphs, we were able to determine what fluorescence threshold works best for each positive control, which does seem to vary depending on the targeted species. So for example, boa constrictor's fluorescence threshold is around 129, and North African python's threshold is around 62. So at this point, we know that our digital PCR multiplex assay works with these individual snakes, and it does not cross-amplify them. But when we take this out into the field and we are actually going to detect environmental DNA, we might have multiple snake species in the same sample. So there might be a boa constrictor and a Burmese python out in the same water. And when we collect that water, we want to be able to detect both of them at the same time. So we needed to make sure that this assay works for a mix of DNA in the same sample. So to sort of emulate this, I took the total DNA from each species of snake and I combined those DNA extracts in different combinations. So either groups of 2, groups of 3, or all 4, and then tested these on our digital PCR using the multiplex assay. And here are the results for this. So on the y-axis is the number of copies per microliter estimated by the digital PCR. And on the x-axis is all the different combinations of snake species that were included. Now, there is a lot happening in this graph, but what is important is that the results are consistent with what DNA was present in the sample. So when the sample was just boa constrictor DNA mixed with Burmese python DNA, those species tested positive, the rest tested negative. Same thing if it were 3 species combined in the same sample. The 4th one that was not present tested negative, which is good. And when all 4 species were in the same sample, again, all 4 species tested negative. So at this point, we know that our multiplex assay works on these controlled DNA extracts that we obtain from muscle tissue, even when they are all mixed together, even when multiple species DNA is in the same sample. But then we wanted to make sure that our assay actually works on environmental DNA. Is it able to test those small concentrations, and is our form of obtaining that environmental DNA and extracting it going to work for the digital PCR? So to do this, we first started with what I call mock eDNA samples. So to generate these, I took that muscle tissue DNA extract from a Burmese python, and I mixed different concentrations of it with a liter of water. And then for processing those environmental DNA samples, we use a vacuum pump and filter the water through a membrane, a nitrocellulose membrane, and then extract those membranes using a kit that is specific for water samples and test them on the digital PCR. We also realized at this point that eDNA might not occur as just strands of DNA floating around in the water. So it's probably going to be bits of cells, bits of animal tissue, possibly. So we wanted to also generate a mock form of this. So to do this, I actually took Burmese python muscle tissue and weighed out different amounts of it and pulverized that tissue, ground it up, mixed it with a liter of water also, and processed those the same way using the vacuum pump, a liter of water through that nitrocellulose membrane, extracted those, and tested them with our digital PCR assay as well. So here are the results for just the DNA extract mixed with water. Here is the range of concentrations that I tried. So anywhere from 4 micrograms of the total DNA in a liter down to 50 nanograms in a liter, we're testing positive with our digital PCR assay. We saw that with 1 nanogram mixed with a liter of water, that resulted in a negative. So we believe that our limit of detection for at least DNA extract in water is somewhere between 50 nanograms and 1 nanogram. Now, for the muscle tissue mixed with water, it's a bit of a different story. So if you notice, these 4 concentrations here have asterisks, and that's because the muscle tissue actually clogged our filters to the point where I could only process half of the water that it was mixed with. But regardless, whether it was a half a liter of water or a full liter of water with these smaller concentrations, we were getting oversaturated chips or arrays. So we realized that this is probably not the best representation of eDNA out in the field. So this is very likely to be a very over-exaggerated version of environmental DNA. So we had to move on. So the next thing that we tried was what I'm calling controlled eDNA samples. So to do this, we actually took a live Burmese python and we placed her in a toad of water. And then we sampled from this water, a liter of water, 5 minutes, 30 minutes, 1 hour, 2 hours, and 3 hours while she was in the water. We also later on had a Burmese python hanging out on a patch of soil. And we had the snake on this patch of soil for a few hours. We removed her and then sampled from that soil 24 hours later, 1 week later, and 2 weeks later. We then combined that soil sample with a liter of water and used the vacuum pump to process it the same way. And then we extracted those filters and tested them on the digital PCR once again. And here are the results for this. So with the water samples, of course, our baseline with no snake in the water was 0 as expected, but even at 5 minutes of the Burmese python being present in that water, we're getting positives. And all the way through the duration of these experiments, we are getting positives for anywhere between 5 minutes and 3 hours of the snake being present in that water. Now for these soil samples, we're also getting positives at all of these time points. So 24 hours later, a week later, and 2 weeks later after the snake was removed. Although you can see the concentration of copies per microliter does seem to decrease over time. Now, whether that decrease is due to DNA degradation, this soil was outdoors in a outdoor enclosure, and it could very well, it was exposed to the elements, and of course, time can lead to the DNA degradation. But it's also possible that we saw a decrease in concentration just because we were simply removing more and more eDNA as we repeatedly sampled this area. So at this point, we were confident that our digital PCR assay works for these snakes. There's no chance of cross-amplification, minimizing our risk of false positives or false negatives. And we know that it should work with environmental DNA samples due to our controlled experiments. So this demonstrates the first tetraplex digital PCR assay for snakes, as far as we know, and it could potentially be used for early detection for range expansion of these snakes. So in the figure to the right, this represents all the different ranges of these constrictor snakes. So the large green range here is the range for the Burmese python. The purple is for the boa constrictors. The yellow triangle here is the rainbow boas, and the small blue range here is the North African pythons. You can see there's a lot of overlap happening between these different constrictor snakes. So it's very likely that it is clear that they are often occurring in the same areas. And so this assay is really versatile in that it can be used to detect the multiple of these species at the same time. We can also use it to verify sightings. So should someone see a Burmese python off the side of the road, we can use this as a tool to aid in making sure that that snake was actually present or not. We can also use it to assess removal efforts. So when these managers or researchers remove snakes from these areas, they do need to go back and make sure that they removed all of them. And this is potentially could be used as a tool to help ensure that they removed all those snakes. It can also be used to aid in decision making. So it can help researchers and wildlife managers decide where to prioritize their efforts based on the amount of positives that we're getting from one area versus another. So we have done all of this work to get this assay ready for snakes. That I have talked a lot about just now, but we're also doing the same thing for other invasive species here in South Florida. So we're working on doing the same thing for these two species of invasive fish, the Asian swamp eel and the bullseye snakehead. 3 species of crocodilian, so the invasive spectacled caiman, but also the American crocodile and the American alligator, which are 2 native species that we want to keep track of. 2 invasive monitor lizards, which are the Asian water monitor and the Nile monitor. And 3 species of tegu, the Argentine black and white tegu, the gold tegu, and the red tegu. Here is our progress on getting all of these assays up and ready. So I have explained with these snakes, we have done our assay design. We've ensured there's no cross amplification occurring with the different species targeted. We've obtained multiplex data when all the primers and probes were combined. And we ensured that this assay works with controlled eDNA samples with the snake in the water or on the soil. So we've done all of those steps also with the fish and the crocodilians. We have yet to do the controlled eDNA experiments with the monitors and tegus, but that's up next. And we are, at this point, pretty confident that this assay will work for these eDNA samples. It's just a matter of optimizing our methods for actually collecting these samples out in the field and making sure that our way of processing these samples for the technology to test them works properly. So here in this figure, this is actually an example of us taking this test out into the field, but for our fish assay rather than our snake assay. So this is a canal that is known to have the invasive bullseye snakehead in it. You can see, this is kind of hard to see, but the bullseye snakehead is here in the picture. And we actually took water samples from this canal and tested them using our fish assay. And we did get some positives out of this canal. So it is a start for getting these assays ready for actual field collection and use for wildlife managers and researchers. Quickly, I would like to thank the Vector Entomology Lab, which is the lab I belong to. My PI is Dr. Brian Bader. Our funding sources are the South Florida Water Management District and the Invasive Science Research Institute at UF. This was very much a collaborative project between our Vector Entomology Lab and the Croc Docs, which are the Wildlife Conservation Lab at the Fort Lauderdale Research and Education Center. So these folks are the ones to actually obtain the invasive species for our positive controls. They are the ones to handle the live species for our controlled eDNA experiments. They're the ones who often go out and sample the water for our field testing. We also received positive controls, the muscle tissue from the Florida Fish and Wildlife Conservation Commission. If you have any further questions for me, my contact information is here. Thank you very much for listening to my presentation, and I'm happy to take any questions.
Thank you very much. That was very interesting. It isn't quite what I expected. I thought you had a magic thing you carried around with you into the field, and it immediately told you everything, but you have to work a lot harder. So my first question is, you kept talking about PCR. Please define PCR.
Yes, absolutely. So PCR stands for polymerase chain reaction. Long story short, it is a way for us to take a specific section of DNA that we're looking for and replicate it a bunch of times or amplify it, make many copies of it, so that way we're able to actually see if it's there or not. So actually, if we go back to the slide, it was many slides ago. Oh, here we are. So you can see many different forms of PCR. We chose to use the digital PCR for this process because it's super sensitive to these small amounts of DNA compared to conventional PCR or quantitative PCR.
And did you find an area in your sampling where there were no Burmese pythons?
So we haven't really taken this out in the field for testing for the snakes yet. We've done it with the fish because they are the field sites for these fish are conveniently nearby. But I will tell you some of the further experiments that we've done for testing this on snakes. We actually had a snake crawl along some cattails because there are concerns that there is Burmese python presence in some natural areas in Chi, and they believe that the snakes are crawling along sawgrass. So we actually took a Burmese python, had it crawl along some
Oops, sounds like we
up.
lost you.
Oh no.
No, you're back.
Oh, good. So I was saying there is concern that there might be some Burmese python presence in Loxahatchee in these natural areas, and that they might be crawling along sawgrass. So we wanted to first make sure that we were able to possibly detect these snakes from plant samples. And to do so, we actually had a controlled experiment in an outdoor enclosure where we took a Burmese python, had it crawl along some cattails, and then tested those cattails for Burmese python DNA. And they actually did come up as positive. So at some point, we are going to take sawgrass samples and see if we get Burmese python DNA out of them, see if they are actually present in those areas or not.
Thank you. Any questions from the audience? I have one.
This is quite a fascinating project, especially for your PhD.
Thank you.
Good luck with it. I have a couple of questions. First is the genome of these snakes figured out? Is it known, the total genome?
I will be honest, I am not sure. I'll have to check. For this assay, I know that the mitochondrial genome, I believe, is figured out because when we designed this assay, we used the CO1 gene for nearly all of the different species that we work with. So for all 4 species of the constrictor snakes, we used the CO1 gene, which is a mitochondrial gene.
Okay, okay. Next is, as Walter said, I thought you would be using this in the field to find the snakes, but you had
I
only
wish.
this to see whether there is a population of snakes in that area. So since the DNA persists for, you showed like what, 2 weeks on the surface,
Oh,
was it?
I think I might have lost you.
Yeah, you did. We've lost you. What happened? Okay, can you hear me now?
Oh, yes, I can hear you.
Yeah. So I was saying that since the DNA seems to persist in the environment for several weeks, if a snake passes over it, how exactly do you think, how precise would your localization be in terms of practical terms, the usefulness of the technique?
Yes, so we still have some work to be done to determine exactly how long the DNA will persist. At this point, it will probably be good for assessing if an animal is present in a new area, but we need to do further assessing to see how long the DNA will truly last out in these elements. We have done some experiments outside in a pond to sort of assess like the snake presence in water in a more natural sort of setting. And with those samples, we would only get positives for like the first day or so of the animal being in that water. So it could very well be that in a less controlled environment that the DNA doesn't persist as long. And it could also vary depending on the environmental conditions as well. So that is some other factors that we need to still assess for before we get this ready for field use.
Okay, one final question. The Everglades, where these snakes are, it's all swamp area. How do you know where to sample your water, where to get the water from? You have specific grids or something? I mean, how would you do it?
So that is not a question that I am fully prepared to answer because I am not the one actually going out into these places to find these animals. That would be more so a question for my colleagues, the wildlife conservation folks, because they're the ones actually going out into these
Okay. Yeah,
research areas.
yeah, okay, okay, thank you.
Thank you.
Any other questions here in the audience? Is there anything on Zoom? Nothing on Zoom. So thank you very much for appearing remotely. And we had a good, successful run through the whole thing with very minor glitches. So thank you very much. We appreciate it. I will be sending you and your advisor, thank you very much, kind of an email. Next week, we will have Ray Goldwire's grandson will be here. Ray Goldwire, as you know, is a resident of Okemuk. His grandson has done research in a river in Africa, and we're going to find out the beneficial uses of hippo poop. So come again next week, and we will have a good turnout, we hope. And thank you very much again, Melody.
Thank you for having me.